Hi, my name is Elvira Fortunato and I'm the director of CENIMAT, the Materials Research Center. Invisible and DigiSmart are the two advanced grants I got from ERC, the first in 2008 and the second last year, and both of them have in common the use of sustainable materials and green technologies to be used in disruptive electronic applications. With the Invisible project, it was possible to consolidate the area of transparent electronics at CENIMAT as well as in Europe by setting up one of the best teams in the world and by promoting the reinforcement of state-of-the-art high-tech infrastructures. Also, we are very proud because Invisible project was selected by ERC as a success story. With DigiMart, the new project, we want to make a revolution in the way our electronic devices are fabricated by using digital platforms as well as the implementation of a new concept based on integrated functions, which is opposite to integrated circuits. I would like to thank ERC for promoting the scientific excellence and believing in us. Thank you and welcome to Nova University.
My name is Mariana Pinho. I'm the head of the Bacterial Cell Biology Lab at ITQB from Universidade Nova de Lisboa. So the ERC consolidator that I got last year, which is titled KronosAntibiotics, because it aims to look at what happens to bacteria over time and use that knowledge to find windows of opportunity during which bacteria are more sensitive to antibiotics. So the project works with a bacteria which is pathogenic called Staphylococcus aureus. It's important because it causes a high number of infections resistant to antibiotics in the hospitals as well as in the community. And the project has two major aims. One, which is mostly fundamental science, is to understand the regulation of the bacterial cell cycle. What does this mean? So we are not the same when we are babies or teenagers or adults. So the bacteria are also not the same during their cell cycle. And so there are different periods of the cell cycle during which we know that bacteria are more susceptible to antibiotics, and we want to learn this information. We also want to learn how is the regulation of this process, so understand how bacteria transition from one stage to the next. The second big aim of the project, which is more applied, is to construct Staphylococcus aureus strains which detect the presence of different antibiotics. So this is the idea. Let's say you go into a very warm room. You'll have a response, which is probably sweating. So bacteria also respond to changes in the environment, but also to compounds that inhibit certain metabolic pathways. So, for example, penicillin inhibits the synthesis of the cell wall, which is kind of the exoskeleton of bacteria. So these responses to antibiotics are mediated by changes in gene expression. So there are genes that are turned on or off in the presence, let's say, of penicillin. And gene expression is regulated by the promoters of each gene. So this is a bit of DNA which is upstream of each gene that turns it on and off. So if we learn that, if we know which genes are turned on in certain conditions, we can place a gene encoding a fluorescent fusion, so a fusion that emits fluorescence, green, red, etc., under the control of a specific promoter. So the idea is when you now add penicillin, the bacteria will become fluorescent and then you can identify. So if you have a lot of these reporter strains, you can detect compounds that inhibit cell wall synthesis, that damage DNA, etc. So you can look for new compounds with antimicrobial activity, and you can also learn how existing compounds work. So these are the two major aims of the project. In my opinion, the ERC is the best funding mechanism for science in Europe because it's one of the few that funds fundamental science to a high level so that we can actually pursue relevant questions with a lot of freedom. For our group, it was absolutely crucial to maintain and renew the team of excellent people that I had assembled with the first ERC, as well as to buy state-of-the-art microscope, a super resolution microscope that is crucial for us to remain doing science at a competitive level.
My name is Isabel Ferreira. I won a Consolidator grant in 2015. The name of project is Capture PV, and the idea is capture the thermal and photovoltaic energy and produce an autonomous device that includes also the storage of energy. So this way it is possible to develop an autonomous device that is able to supply energy all the day. This grant was very important to develop this work, and for that we bought different equipments, and also was possible to hire different people, postdoc, PhD students, and also master students. Without them, it would not be possible to develop this project. So I'm grateful to ERC for this consolidator grant.
So hello, my name is Luís Pereira, professor at FCT Nova, and I will explain what we are doing in NewFun Project. So NewFun Project is all about establishing the foundations to move from something like this to something like this. So the idea is to create electronic devices on paper, and for that we need to develop materials that are able to do so. We are working at different levels at this project, so starting from the substrate itself, so starting to make paper that is able to support the electronic systems that we want to make, and this means that we need to modify the paper from the beginning, so from selecting the right fibers and to make the proper treatment to those fibers in order to get the right properties for our substrates. Next, we need to develop the materials that we want to deposit on paper to create the electronic systems. And so we are using cellulose-based materials as well to develop inks, functional inks, that we use to create transistors, sensors, and other things that we need to make the electronics on paper. One example is to use modified cellulose fibers. So this is a suspension of cellulose fibers that we use to create cellulose membranes that can be used as dielectric in transistors. Another thing is to develop also inks that also contain cellulose to modify the viscosity of those inks, and that we use then to print. We are also making some new things, that is to go to the minimum size of the cellulose fibers to get cellulose nanocrystals that present very nice optical properties that can be used as a photonic filters, and they present colors, so we can select some colors and use this selection to create some specific sensors that can also be integrated on these platforms that I told you about. So this is the idea behind the project. We are doing pretty well, so we have already some nice publications, and I hope that in the near future you can really start seeing things like this in concepts like Internet of Things. The NewFun ERC grant was very important, not only from the personal point of view for my career, but also because it allowed to establish the lab that you see here, and also to hire the people to establish a team that is doing this fantastic job.
My name is Cristina Silva Pereira, and I'm working at ITQB Nova, where I'm leading the ERC Consolidator grant entitled Mymessage. So the vision of my grant is to develop new antifungal therapies inspired through the mimesis of plant defensive polymers. What are these plant polymers? So they are lipid polymers that build a defensive barrier in most plant tissues, and they have evolved and they were acquired by plants to allow the protection to the UV exposition, the protection to biotic and abiotic stresses. And I believe, and that has inspired the vision of the project, I believe we can dissect the secrets of these polymers to actually identify scaffolds that we can use to develop antifungal therapies. It's also important to highlight why we need new and efficient antifungal therapies. Fungi kill nowadays about 2 million people per year, not healthy people. They provoke systemic infections in people that have immunocompromised conditions, and as superficial infections, they are able to infect 1/4 of the world population. Well, the vulnerable population can be shown by the fact that diabetic people, for example, taken as example, have conditions that hinder the healing of their skin, and skin injuries are an entry door for infections with fungi, and they constitute about 70% of the world population. We have a very limited number of antifungal drugs that are used in the clinical setup, and we are witnessing the rise of the number and the diversity of strains capable to provoke these opportunistic systemic infections. We are observing the rise rise of the number of fungi carrying antimicrobial, antifungal resistance, multi-resistance, and also the rise of the most vulnerable population. And all these factors are still escalating. So this is a very multidisciplinary project because it is bringing chemical topics close to the biology. So because of this, we are using a lot of NMR, solid state techniques, X-ray, but at the same time we are using a lot of genetics and microbiology and biology techniques as well. And so this grant has allowed me to bring people with this expertise to my team and also to acquire equipment that allowed us to become more autonomous in handling some of these experiments. Overall, it also promotes the team and the quality of the team and the visibility of the team across the country, inside the Institute, but internationally as well. And I'm very, very grateful to have had this opportunity and to be leading this ERC grant.
My name is Cecilia Roque. I'm a bioengineer and I'm an associate professor at the chemistry department at the School of Sciences and Technology at Nova University of Lisbon. So today I'm going to talk to you about an ERC starting grant that was awarded to my lab, and it's called SAND. It's called SAND because it's related with artificial olfaction, which is a field that aims to mimic the natural olfaction systems. So in this project what we are trying to do is to build an electronic nose that is going to be used to smell bacteria. In particularly, we are very interested in bacteria involved in human infections, and for that, the great novelty that we actually introduced in this project were the materials. So we have materials that we developed in our lab that can yield optical and electrical responses in the presence of volatile organic compounds. So while bacteria are growing, they actually release, as part of their metabolism, volatile organic compounds, and they act like as a fingerprint for different bacteria. And this is how we can actually distinguish them. So having been awarded this ERC starting grant was a unique and a great opportunity to our lab. Because we managed to assemble a really fantastic and very interdisciplinary team of people, because we have from people working in microbiology, chemistry, biotechnology, we have material scientists, we have electronic engineering, we have people working with signal processing. So this is really something that would have never been possible without a grant from the European Research Council.
My name is Ana Rita Duarte. I am Associate Professor at the Chemistry Department of Faculdade de Ciências e Tecnologia from Universidade Nova de Lisboa, and I'm the PI of DESOLVE, when solids become liquids, natural dipiotetic solvents as green solvents for chemical process engineering. But what is DESOLVE? DESOLVE is a new generation of solvents, green solvents that are able to replace the existing ones with the benefits of meeting the principles of green chemistry. They are non-toxic, they are made of naturally occurring molecules, and therefore they are biodegradable. This is our lab. This is where currently nearly 10 people work here every day, and this was what the ERC grant offered me, the possibility to further explore these solvents for different applications. For extraction of high added value products from agroindustrial wastes, the possibility to study biocatalysis, reaction processes using enzymes that have higher yields of production, and also to study these solvents in therapeutic applications, in different applications for disease therapies like tuberculosis, like anti-cancer treatment, but also in their antibacterial and antimicrobial properties.
My name is Pedro Barquinha, and I'm an associate professor here at the Material Science Department. And I'm here to present you the ERC starting grant that I'm running since 2017, which is called TREND. So TREND is all about creating a nanoelectronic platform that can be flexible, transparent, high performance, and at the same time can be even self-powered first we need to make nanotransistors and nanocircuits. But the thing with that technology is that it's high temperature and it's not scalable to very large areas. So if you want to do something in the square meter range with nanotransistors, there's no technology for that yet. And then the other thing is you need to create some ways of self-powering these nanocircuits. So to make these two major advances happen, we are using as a basis oxide nanostructures, particularly what we call by oxide nanowires. So these are like 1D structures, and these are great for electronics because in a structure like this, you can essentially confine the electron movement to a specific direction. And you can also imagine, for instance, for the energy harvesting applications, that you can deform the nanostructure in this axis, and so you can get energy out of that. What we are doing in terms of innovation on that is using what we call a multi-component oxide approach. So we know this from microscale. We put materials like zinc oxide, tin oxide, and so on working together. But now at the nanoscale, this brings a completely different perspective into the material science field. So what we can do with this, take for instance our favorite material into this, the zinc tin oxide. You can put together zinc and tin oxide, and you can have material that is suitable for different applications, let's say a high mobility semiconductor or a very good piezoelectric material to get energy. And these always based in the same material system. It just depends on the way that you synthesize it and the phases of the material that you obtain. Okay? So this is very important because it alleviates quite a lot the work in terms of integration, integrating devices that do this, do that, and do that. So all of them are essentially based on the same kind of material. So it's very good for integration, and of course, it reduces costs. And then the thing comes to how can we use these nanostructures? So that's why we are here in this lab. So essentially the machine that is behind me is what we call a nano-imprint lithography tool. So what this allows us to do is to precisely define where we want to create these nanostructures. So if we want to, let's say, to create a network with 1,000 nanowires, or if we want to create a network with 10 nanowires, this will have completely different properties, and this machine allows us to do precisely that. Okay? So machines like this would only be possible to acquire with a grant like this, so it's a very expensive tool. And even more important than that, this grant helps you to set up your team. It's also very important because it allows you to get together with really excellent scientists among the entire world. And for instance, I'm involved within the framework of this ERC grant in a network which is called Young European Material Research Society, which I'm one of the founders. And essentially we try to put together these ERC laureates, for instance, to speak to audiences and to tell them how great these grants can be to trigger your career and what are the perspectives that you can have to have scientific independence, regardless if you go through an academic or industrial path.